So the contractile proteins each have a unique structure. The regulatory proteins also have an interesting structure, which we can take a closer look at here on this slide. Now, one of the really annoying things about this topic is the general lack of creativity when naming things. So tropomyosin and troponin sound the same, but they're two different regulatory proteins. Tropomyosin and myosin sound the same, but one's a regulatory protein and one is a contractile protein. So apologies in advance for the naming. But tropomyosin is a filamentous rope-like protein. In this diagram, it's the teal protein running alongside actin, which is in yellow. Troponin, on the other hand, is a smaller protein made up of three subunits, TNT, TNI, and TNC. TNT binds to tropomyosin, TNI binds to actin, and TNC binds to calcium. You don't have to remember what each of these subunits binds to, but it can be helpful to remember that troponin is the one that has three subunits that bind to different things. And specifically, that troponin is the calcium binding protein because that's important for muscle contraction regulation. Now, in order for muscles to contract, filaments have to be able to slide. And that means there must be binding between actin and myosin. This binding between actin and myosin is often referred to as a cross bridge. So in order for contraction to occur, cross bridges must be able to form. Tropomyosin regulates binding by blocking myosin's binding site on actin. So when tropomyosin is present, which is the situation that we have when there's low intracellular calcium, it's physically impossible for cross bridges to form because the myosin can't access that binding site. When the calcium concentration inside the cell increases, the calcium binds to the other regulatory protein, troponin. And troponin then has a conformational change that allows it to move the tropomyosin out of the binding site. This makes it physically possible for cross bridges to form. Now, all this means that skeletal muscle, actually all types of muscle, require a calcium signal to initiate muscle contraction. The other requirement for muscle contraction is energy. In the last unit, we talked about how important ATP is for driving active transport and for maintaining concentration gradients in all excitable cells. It's important in muscle too. But ATP is also used in other ways inside of skeletal muscle. In fact, skeletal muscle is one of the major consumers of glucose inside the body. And that's because skeletal muscle fibers have a really high concentration of mitochondria, which allows them to meet these great energetic demands. Now, one example of skeletal muscle using ATP is for the polymerization of G-actin into F-actin. That process is ATP dependent. But the most important role for ATP is in the regulation of cross-bridge formation. So the next two slides are basically describing a cycle of ATP hydrolysis that takes place on the myosin head. And this is the second thing that determines whether interactions between actin and myosin are physically possible. Because this is a cycle, we could really start anywhere, but the place that I'm going to start is when ATP binds to the head of myosin. So when ATP is bound to the myosin head, the myosin has a very low affinity for actin. And this means that cross bridges can't form. It also means that any existing cross bridges are essentially broken by the binding of ATP to myosin. But fortunately, this is a short-lived state because the myosin head is enzymatic and it can hydrolyze ATP into ADP and phosphate. ADP and phosphate, the phosphate being represented by pi, stay bound to the myosin head. And when they're bound, the myosin head twists into a high energy conformation and binds to actin. This state is often referred to as the cocked conformation because it's a high energy state and it's very similar to cocking the trigger of a gun. In the next step, the phosphate gets released and that allows the myosin head to stay bound, but to untwist and to move into a low energy state. And as it does that, it pulls the actin one position closer to the center of the sarcomere, or one position closer to the M line. In the low energy state, the ADP gets easily replaced by ATP. That allows the cross bridges to break, and this whole cycle can repeat a second time. So to quickly summarize all of the molecular events of contraction that we've talked about in this part of the unit and see how everything fits together, remember that this story starts off with calcium, because without calcium, binding is physically impossible. The tropomyosin is attached to actin and it's in the way. The myosin doesn't have access. So when the calcium level inside of the cell increases, that calcium can bind to the other regulatory protein, which is troponin, specifically to troponin C. That troponin will then create a conformational change that will pull the tropomyosin out of myosin's binding site on actin. This makes binding physically possible. Once the binding is physically possible, now that ATP cycle can kick in that we just talked about, which will ultimately generate the power stroke and allow the filaments to slide one position closer to the M line. And then that whole process can repeat as long as the calcium levels inside the cell remain high.